The human brain is the most complex structure in the known universe. The largest region of the brain is the cerebrum, here depicted in aqua. And in other videos, I've gone through the other regions of the brain, such as the medulla, the pons and the midbrain, which form the brainstem, the diencephalon, which is here and also forms the same part of the embryonic forebrain as the cerebrum, and the cerebellum. So once again, as the embryonic brain forms, it forms as three separate lobes, which two of which then subdivide. And so here you have the prosencephalon. This will end up forming the cerebrum and the diencephalon, the mesencephalon, which will form the midbrain, and then the rhombencephalon, which will then form the cerebellum, the pons, and the medulla. And so one of the significant things about mammals, and then even more so of primates, even more so of apes, and then even more so of humans, is the enormous expansion of the cerebrum, so much so that it then covers the midbrain as it folds back over that. And so once again, this is the largest part of the human brain, and one of the two regions developed by the embryonic prosencephalon. Just to look at some images before getting back to the videos, here you see a brain of a sheep, and notice that in life it would be covered by three layers of meninges: the outer dura mater, the tough stuff, which is made of the collagen, which gives it the white color, the arachnoid underneath that, and then the innermost pia mater. And that when we look at the brainstem, notice the white matter which we see there. Notice that it's a bit lighter in color than the cerebrum. And here you can see some of that pia mater, that thin film that covers the folds of the cerebrum here. And so the most of the brain's gray matter is in the two largest regions, the cerebrum and cerebellum, while the brainstem is primarily white matter. As I covered elsewhere, although the cerebrum is largest in terms of volume, in terms of neurons, the granule cell layer of the cerebellum is where neurons are most densely packed. And these neurons here compose about half the neurons of the whole brain. And so the cerebellum has a number of neurons far greater than its size might lead you to believe. As we look at the cerebrum, once again, it's big. It is the most complex of the brain regions. It's the site of our conscious thought, much of our personality, etc. And so therefore, just how do we speak of something so large and complex? Well, we're obviously going to anatomically divide it into regions. And there are four lobes of the cerebrum, which can be recognized based on the bones of the skull, which lie over them. So there's a frontal lobe, a parietal lobe, a temporal lobe, and an occipital lobe, named for the frontal, the parietal, the temporal, and the occipital bones, obviously. And so here you can see in sheep, as a mammal, a much larger cerebrum than, say, a reptile would have. That's one of the things that mammals expanded. But notice that it is nowhere near as expanded as we see in, say, in humans. Now, there are these two lateral halves of the cerebrum. And then even in the sheep's brain, you can notice that there are these folds and fissures. Now, since mammals change gradually over the time, many of these folds and fissures are lineage specific, so that what we see in primates would vary from what we see in the sheep. A fold is known as a gyrus, and the groove between folds is known as a sulcus. So one of the things that strikes us most about the cerebrum is that it is not smooth the way the cerebrum of a reptile would be, or even some mammals have a smooth cerebrum. That was the primitive condition. That mammals, whether it be this sheep or whether it be then the human, you can see that there are these folds here. Now, we can name some of them. I'm only going to name two just as part of this introductory lecture, but we can name these. So for example, we could name a central sulcus, which would separate the frontal lobe from the parietal lobe. And then functionally, this is important because the gyrus, which is in front of the central sulcus, the precentral gyrus here, that's where we have that motor cortex, the motor map of our body, as I'll get to. While behind the central sulcus, the post-central gyrus, this is where we have a sensory map of our body. And so if you're trying to identify the parts of the brain, which are significant, these grooves and folds can be important in their just as a point of reference. So this central sulcus separates the precentral gyrus from the post-central gyrus. This longitudinal fissure splits the left cerebral hemisphere from the right cerebral hemisphere. So once again, back in the sheep's brain, here's a longitudinal fissure, which is going to separate the cerebrum into two different hemispheres. Now, for the most part, what's going on in the left half of the brain is similar to what's going on in the right, but there are some differences. So, for example, the left hemisphere does a bit more with language. That's where the motor speech center is located. So reading, writing, speaking are more centered in the left hemisphere, as are tasks such as logic. Whereas things like the appreciation of art and music, map reading, and the ability to rotate something, in three-dimensionally in space, or to orient in 3 dimensions, that would be a bit more on the right hemisphere. And so if someone were to suffer a stroke, there might be slightly different expectations as to whether they damaged a specific area on the left hemisphere and on the right. So while there are similarities, once again, the left and the right hemispheres do specialize a little bit to some degree. Now, notice that there is white matter here, and that white matter is composing the majority of the interior of the cerebrum. Now, these axons do lots of things. They can go in different directions, and I'll go back to a video in a second. So here you can see on a human cerebrum all of this white matter here containing axons, and here the white matter was stained black. These are the axons here. So these axons are important because, number one, that would allow, say, a neuron in this gyrus to communicate with a neuron in a nearby gyrus. So we would call that an association fiber if it went from, say, one gyrus to another gyrus. Some of these axons allow the left hemisphere to communicate to the right hemisphere. So notice here there's what's known as the corpus callosum, which is a big band of axons which unites the two hemispheres and allows them to communicate. So you could hold something in your left hand, which would then be analyzed in the right half of your brain, but then you could talk about it, knowing that it's the left half of your brain which is going to initiate the speech. Well, how can the left half of the brain, which is going to, of the cerebrum, which is going to do the talking, know what is being processed in the right half of the brain, telling us what the left hand is holding? Because this corpus callosum has fibers which will go back and forth and allow the two to communicate. And so we can see uniting the two cerebral hemispheres is this big band of white matter. Notice it's lighter in color than the surrounding area. This is the corpus callosum here, once again allowing the two hemispheres to communicate together. If one were to cut the corpus callosum, and sometimes in surgery that needs to be done to treat, say, a tumor or something, then one has what's known as a split brain phenomenon, where you could hold something, you could hold something in your left hand, which would be processed in the right half of your brain, and you would know what it was because you could find it again in a box, let's say, but you couldn't name it because the two halves of the brain would not be communicating if these two were cut. Now, there are also additional places where there are fibers which are commissural fibers which cross the midline. There's an anterior commissure and a posterior commissure, but they are smaller. The corpus callosum is the major contact there. So these axons include association fibers going from one fold to another, to the commissural fibers, which go from the left hemisphere to the right hemisphere. And then also there are projection fibers. And when I get back to the videos, I'll mention this again. So for example, the thalamus has, an incredible number of fibers which are communicating with the cerebrum and back. And then the cerebrum will send some fibers to the thalamus, which then go back to the cerebrum. But the cerebrum connects to other regions of the brain as well. So the thalamus, the brainstem, the cerebellum, these are called projection fibers. So the white matter here includes axons, which are going in different directions. Association fibers, neighboring gyri, commissural fibers, left to right hemispheres, projection fibers going to different parts of the brain. When you look at the folds, the gyri, the reason for them is obvious, because the gray matter is where the soma are, and the more soma that a brain has, the more complex processing that it would be capable of. Now, there are soma deep inside the cerebrum. There are what are known as the basal nuclei of the cerebrum. But that is not the majority of the gray matter of the cerebrum. Notice that the majority of the gray matter of the cerebrum is in the outermost couple millimeters in what's known as the cerebral cortex. All right, so just like the cortex of an apple, it's just that thin outer red superficial covering. Here, the cerebrum has a very thin cerebral cortex, but it is here where most of the gray matter is. So this brain is not stained, and you can see the difference between the white matter where the axons are and the gray matter where the soma are. But then one can see this as well, that the gray matter is this light yellow. And notice that the majority of the gray matter is in the cerebral cortex. Is there gray matter deep inside the cerebrum? In these basal ganglia, or these basal nuclei, such as the claustrum, the globus pallidus, the putamen, the caudate nucleus here in the amygdala? Yes, but the majority of the gray matter is in the outermost couple of centimeters, of millimeters. There you can see the amygdala and some of these basal nuclei. Again, and the same thing would be true of sheep. Here you can see the white matter there and the duller grayer color in the cerebral cortex. So this is why the cerebral cortex has these folds. It increases the surface area, which allows so many more neuronal soma to exist, which will then allow for so much more processing. And so the significant thing about the cerebral cortex is, once again, these folds, the gyri and the sulci, which give so many more neurons, and thus then so much more processing ability. And so the key to the nervous system, and certainly then the cerebrum, is just the organization. There's so much information which is stored, organized, so many decisions made. It is certainly not the case that there's just this random scattering of information. And so in previous videos in my series, here on the brain, I've gone through the limbic system, which just bears a little, let me repeat the several items that I've already covered. So if you were to ask, do you have control over your anger, okay, or your hunger, or being thirsty, or being frightened, or sexual desires, etc.? Well, I think most people would answer yes to a degree. So yes, we can control our anger, but it's not as if it's completely voluntary, where we say, I think I'm going to be angry right now. And so functionally, these emotions and drives, I think a lot of people would say, are kind of in a limbus, in the middle between the purely voluntary and conscious, and then the involuntary and unconscious. They're kind of in the middle. And then when we look anatomically, they're also kind of in the middle. So the brainstem is completely involuntary. The cerebral cortex is where we have, our conscious, thoughts and our voluntary processing. And if you ask what is involved in emotions and drives, it's a lot of these areas which are in the middle. So those deeper nuclei. So there's some parts of, say, the diencephalon, like the thalamus and hypothalamus, but then also deeper parts of the cerebrum, the amygdala, the hippocampus, the fornix, the dentate gyrus, the parahippocampal gyrus, etc. And these then, once again, both functionally and anatomically, are kind of in between, the more voluntary conscious cerebral cortex and the involuntary brainstem. As I'll return to later, many of these are critical in memory. So if we're asking, like which areas are consolidating memories and transferring things from, say, short-term memory into long-term memory, it would be the amygdala and hippocampus. So that's obvious for a couple of reasons. You're likely to remember the things that get an emotional rise out of you. All right, so something that scares you, you're likely to remember. Something that makes you angry, you're likely to remember. If you're bored by a specific class, you're probably not going to do well on the exam, because the parts of your brain which are, helping you to form long-term memories are those parts which are activated when something gets an emotional rise out of you. When you're bored, you're not as likely to remember. One of the ways that we know what parts of the cerebrum do is, you can study a patient after they've had a lesion or a brain injury or a stroke. And so it can be, different forms of amnesia can result when one damages parts of the limbic system, such as the amygdala and the hippocampus. Also, in previous lectures, I've gone through how information from the general senses are sent to the cerebrum. Once again, there's a difference between general senses and special senses. General senses are those things we feel lots of places. You feel pain, temperature, itch, tickle, pressure, pain from lots of places in your body. But vision only occurs in your eyes, taste only on your tongue, smell only in your nose, hearing only in your ears. So there are special senses which you only perceive in one place. And then there are the general senses which we perceive all over the place. The general senses ultimately project to the thalamus, and the thalamus then projects to the somatosensory cortex of the cerebrum. This was that post-central gyrus. So if you remember, there's a central sulcus in the middle of the cerebrum, and then there is a fold, a gyrus on one side of it. That would be the precentral gyrus, and then another one posterior to it. So the one that is posterior is the post-central gyrus. And this is what we can call the somatosensory cortex. There is a map of the body located here. And so that if something were to stimulate, if something were to touch the hand, that would be perceived there. If something were to touch, the side, that would be perceived there. If something were to touch the foot, that would be perceived there. The face would be here. So if I have electrical activity, here in my brain, I would say, ah, something's touching my face, something touching my hand. That's the way your cerebrum knows what part of the body is feeling something, because the axons are ordered, and that organization is maintained. Now, in the previous part of this video, the second order neurons cross to the opposite side of the body, so that the right half of my brain is feeling the left half of my body. So when something touches my left hand, that would be, perceived here on the right side of my somatosensory cortex, not here. As I mentioned previously, if you were to look at how space is attributed in the somatosensory cortex, it looks weird. I mean, if this is a map of your body, it's a lousy map, right? Because the head is huge, the hands are huge, the back is tiny. I mean, that's not what you look like. But the size of the area on the somatosensory cortex does not correspond to the size of that body part, but rather the number of receptors which are there and your ability to precisely locate stimuli there. So the bigger the area on the somatosensory cortex, the better you can locate a stimulus there. So you're far more sensitive on your face and your fingertips than you are on your back. In lab, one of the things students can do is, if you close your eyes and you have a marker and your lab partner has a marker of a different color, if they touch with their marker your fingertips and your ventral arm, and then you try to touch exactly the same place, you'll probably come far closer on your fingertip than you will on your ventral arm. You're better at saying, ah, I was touched right here on my fingertip or on my face, than you are your ventral arm or on your back. And that corresponds to the bigger the area on the somatosensory cortex, the better your ability to locate a stimulus there. Now, I'll obviously be getting back to this as we sort of pull all of this together. But once again, someone who has a stroke, you could identify what various parts of the body do. If I have a stroke affecting my somatosensory cortex, I can no longer feel my hand. All right, so this is the area where the general senses from the hand go. If I have a stroke that destroys this area, I can't feel my hand anymore. Now, just as there is a somatosensory cortex, in front of it, the precentral gyrus, we have a motor cortex. And also we have a map of the body there. The face is very large. Once again, the hand is very large. And so the greater the area there, the greater the precision of our control. We have great precision of our control with facial muscles, or the fingertips, as compared to, say, the muscles of the back or the calf. So the precentral gyrus, this motor cortex, that's in front of the central sulcus, that's where, once again, we are going to initiate motor commands. There is a face area, there is an arm area, there is a leg area. And when there's electrical activity somewhere, that doesn't mean you're feeling your face or feeling your hand anymore. It means you're moving a facial muscle. It's where you're moving your wrist, your forearm, your leg. And so these, your brain has never seen your body. So how does it know what to do, or what? Because it has these maps and the axons which come to the cerebrum stay organized and project. And so therefore the electrical activity here will result in a specific movement. In previous lectures, I've gone through axons go from this motor cortex to the spinal cord and what's known as the corticospinal tract, which then carries these voluntary muscle commands. But once again, your cerebrum has more to it than these voluntary centers. Your brain is bigger than your mind, if you like that statement. So in addition to saying, aha, I want to voluntarily move this muscle or turn my neck, if someone were to slam a door, I would jerk my head around as a reflex, because there are other areas which then also contribute to muscle control, not just the corticospinal tract, which forms what we call the pyramidal system, because its axons go through a part of the medulla called the pyramids, these triangular pyramids. We also have the extrapyramidal system. So there's nuclei deep in the cerebrum, in the diencephalon, and the cerebellum. So once again, because these triangular ridges known as the pyramids in the medulla contain the corticospinal tracts going from the motor cortex to the spinal cord, this is called the pyramidal system, and this is where the voluntary motor commands are sent. But we can also send commands to muscle from the cerebellum, from the midbrain, from areas deep inside the cerebrum. So these areas then collectively are called the extrapyramidal system. So they can do involuntary reflexes or even the more complex things. So for example, I could voluntarily decide to start walking, but then have a conversation. So obviously, it was a voluntary command to start walking, but somehow the rhythm of the walking is just being maintained through more or less involuntary ways. So that would be the extrapyramidal system, which would then do that. Now, I have in my playlist of videos, I have videos which are a little more serious, some less serious. And then the less serious ones, the various parts of the brain, they could be talking with an accent. And once again, one of the things I just like my students to get an idea of is that, there's this organization to the cerebrum. And once again, one can see like electrical activity. So if you were looking at EEGs, you could see changes in different brain regions based on activity. All right, so like when you start doing your math homework, you're using different parts of your brain as you do when you're doing English homework, etc. But you could also then see this, and I'm sorry, EEGs and MRIs. You could also then see this with stroke victims. So what's the effect of a stroke? Well, that depends. Which part of the brain did you damage? So here, if I were to damage part of the motor cortex, then I would lose the ability to voluntarily move muscles. Once again, the corticospinal tracts, they cross to the opposite side of the body. So here, my right half of the cerebrum is controlling my left half of the body. So if I were to have a stroke here, I might no longer be able to voluntarily move my left hand. Now, it's a bit more complex because once again, there's those extrapyramidal systems. So if I had a stroke which affected my ability to move my face, if someone said smile for the camera, I might do this, only able to move one side of my face. If someone told me a joke, I might be able to laugh. And you say, oh, you moved that side, do it again. Okay. But there was a difference because involuntary reflexes are governed by extrapyramidal centers. And so it's possible to damage the voluntary centers and not have voluntary control, but still maintain involuntary control, and vice versa, where you might have tremors like we would see in a disorder like Parkinson's, but still have voluntary control where you could stretch out your hand and grab something. You would just have less control of the involuntary motions. And so in some of these videos, I just go through brain regions with the idea that you could then have an idea of, this is where voluntary centers, commands are given versus premotor control. So for example, the motor cortex might say, here do this action. But if something were complex, think of when you learned, how to walk, how to drive a car, how to type, how to play a musical instrument. There's more than just, hey, fingers do this. There's a difference between this and typing. And so you have to, learn, precisely the exact motions, et cetera. And this would involve a couple of areas like the cerebellum, especially if balance is involved. But the area in front of the motor cortex is called the premotor cortex, or the premotor area. And so that would be important as well in memorizing more complex areas. If I were to have a stroke, say, that affected the premotor cortex, I might lose the ability to type. I could still move my fingers, but I would have lost the ability to type. I could learn again, potentially, et cetera. And so, once again, I just have several videos. There's, I use accents involved that, then indicate, if you were to damage this part of your brain, what area might be affected. Let me just go back to, my images once again. Some of those videos, using the accents are a little less serious, but that's what they're trying to get across. So here we would have a motor cortex, a premotor area. Here on the left hemisphere only is where we have the motor speech center. This is not only where we control motor movements, but specifically those motor movements associated with speech. Once upon a time, you learned how to make a B sound. There are specific muscles you have to use to make a B sound, or an M, or a T. And in my classes, I use a South American language, Guarani, and I ask them to say words like Baishapa and things where there are new letters, and I ask them, like, there's a mild curse, anyam and bu, and I ask them to practice that. Well, there's a vowel sound in membu that English doesn't have, so you'd have to practice that before you got good at it. I probably still am only marginally good at it. And so that's what happens in the motor speech center. If you were to have a stroke here, you would lose that voluntary control over speech muscles. You would no longer be able to speak. You would be able to understand speech still. All right, you could understand what someone says because that's a different area of the brain. But the commands to actually these speech centers to make that sound, you would be unable to do that. In addition to the motor centers, there are then areas of the cerebrum which would manage senses. So we have the somatosensory cortex. Here, the very bottom of this would be a taste region. So taste is a special sense. We perceive it from one part of our body, our tongue, and it goes to one part of our brain, while the somatosensory cortex is getting information from the general senses: pain, temperature, touch, itch, tickle, etc. You feel all over your body, and they're all going here, as opposed to taste, which only goes there. So taste is a special sense. Hearing is a special sense. So the auditory cortex is where hearing goes. Only that sense goes there. Vision would go to the occipital lobe. And smell, there is an olfactory area, but you'd have to look on this aspect of the brain in order to see the olfactory cortex. So they would be on the medial surface of the cerebral cortex. So there's a difference between the general senses and the special senses. Each of the special senses has their own area. And there's a difference between what's called a cortex and an association area. There is a difference between saying, I feel something in my hand, I see something, I hear something, I taste something. So that's one skill, but it's another skill to know what it is. I'm feeling a tennis ball, or a can of soda. I'm seeing the face of a friend of mine. I'm hearing the voice of a family member. I recognize that voice. That's spaghetti sauce that I'm tasting and smelling. So in addition to a cortex, we have an association area. So here, if I have a stroke that affects, say, a cortex, I can no longer perceive that sense. I can't feel touch in my hand. I can't see out of this eye. I can't hear out of this ear. I can't taste. So I would lose the ability to sense if I damaged a cortex, like the auditory cortex, the gustatory cortex, or the visual cortex. But then there's also an association area. So if I damage that, I could still see or taste or hear. I just wouldn't understand what I was seeing or like something's in my hand, but I don't know. Is it a tennis ball or a soda can? I'm hearing something, but I don't recognize the voice. Is it my best friend or a family member? I should know their voices, but I can no longer associate that because now the stroke affected the auditory association area, or the somatosensory association area, or the visual association area. I recognize the voice of my friend, but I no longer recognize their face, okay? And so our cerebrum, in addition to having this cortex which receives the raw sensory input, then interprets it. Just a couple more, two more that you can see on this image. This general interpretive area starts to put things together. So we have to do that. So for example, if someone were to say sit here, all right, well, you could say, all right, sit, yes, not stand, sit, here, not there, okay. You can get the definition of those two words, but you have to put them together, sit here, all right? So somewhere you have to be linking the part of your brain that knows what sit means and the part of your brain that knows what here means to understand that sit here is a command that tells you to do this. I joke with my students, you know, like if you're in lab, if your leg was on fire, how would you know that, right? You would see, you know, red flame, you would smell smoke, all right? You would hear your lab partner laughing at you. You would feel heat and pain, all right? But you'd have to put all of those together to realize, oh, the light, the smell, the smoke, the, you know, the this, you know, my leg is on fire. So we have a general interpretive area that starts to make connections between these other regions of the cerebrum, so that, you know, we could, you know, have a more complex meaning. If you have a stroke here, that could be very bad, right? So for example, if I had a big enough stroke, you know, affecting, damaging this area, I could listen to someone talk. I could understand every single word they said and have absolutely no idea what they were talking about, because understanding, you know, each word and then putting it together for a more complex meaning, that's different. Finally, here, there's the prefrontal cortex, just behind the frontal bone. This is where some of the most complex processing in our brains would occur. Much of our personality would be located there. Our ability to understand what is socially acceptable, social norms, distinguishing between past, present, and future. So that, some of our most complex functioning is in the prefrontal cortex. A frontal lobotomy cuts the connection, or this was more common decades ago as a, you know, an often misguided attempt to treat disorders of the mind. It would cut the connections between the prefrontal cortex and the rest of the brain. So if a patient was being very difficult, they had anger issues, et cetera, and you performed a frontal lobotomy, in some sense, it might be considered a cure. They're no longer angry, all right? But their personality would be gone. You wouldn't see much of that person. And one of the reasons they're not angry is if you don't understand the connections between past, you know, present and future, why would I get angry? I couldn't project and see what the future consequences of something might be. So I have a number of, you know, these. Once again, there's accents, they're a little sillier, but it's meant to just kind of walk through what different regions of the brain might do based on what damage to that area might result in. Now, just a couple more concepts in the cerebrum. One is, you know, this has been focusing on the cerebrum like from a largely macroscopic standpoint. But obviously you could look at microscopic changes as well, going back to the chapter on nerve tissue, because obviously we go to school to learn. We want to change our brains. We want our brains to be different after the class than it was before. So obviously at some point we are changing synapses, all right? And if you were going to try to you know, work on study techniques, how do I remember better? So like a good study technique or a synapse that we're using all the time, we would change it and it would then be different. So there would be, say, more synaptic knobs that would release more neurotransmitter, or there might be a different type of receptor that would form at a frequently used a stimulus called an NMDA receptor. Or if it's someone that you see all the time in your class, then as you're approaching class, you already might be releasing some neurotransmitter, meaning that, you know, it would take less of a stimulus to fire. The point is that one of the interesting things going on in the cerebrum is, you know, the cellular changes and the microscopic changes, which are being used at synapses. You know, so you know, how we study and whether we use a synapse a lot, you know, it would, you know, determine, you know, whether we how we remember things. If something got a great emotional rise out of us, so remember that, as part of the limbic system, like that's why the alien is there. So parts like our amygdala and our hippocampus not only would react emotionally to something, but they're also going to transfer memory. So things which get an emotional rise out of us, they are much more likely to be remembered. It's more anatomy and physiology 2 classes as opposed to anatomy and physiology 1, which worry about the blood vessels going to our brains. But we do have arteries which send oxygenated blood to our brains. There's the common carotid, which forms so here you can see the common carotid, which would then branch into the internal carotid, going through the carotid canal of the temporal bone. But you would also then have another source, the vertebral artery, which then goes through the foramen magnum. And together this forms an arterial circle from which arteries go to different regions of the cerebrum. Deoxygenated blood would then go back through a jugular vein. So, at some point that's important to study the blood flow to the brain. We also then have spaces in our brain where cerebrospinal fluid passes. So there are capillaries, which are specialized, in areas called choroid plexus. And from here, we form this filtrate, this cerebrospinal fluid, which will then help to support the weight of our brain, help to regulate the concentration of ions, et cetera, around this, even transport some messages. So oxytocin has multiple ways that it can get from one area to another, but one of those is through the choroid plexus. And if you remember when we talked about glial cells, one of the forms of glia are these ependymal cells, which are ciliated. So they have cilia, which can help transport this fluid that goes through the nervous system. So we make this cerebrospinal fluid at specialized areas called choroid plexus. And then this will then flow through ventricles of the brain. I know it sounds odd to think that you have spaces in your brain, holes in your brain, for example, but our nervous system forms as a hollow tube. So here you see an embryonic brain forming, I think of a frog. So notice that it's hollow. So the brain is hollow when it forms, the spinal cord is hollow. We can still see this in the central canal of the spinal cord. There's a pig spinal cord forming. Notice that it's hollow. So the central canal is the leftovers of that space in the spinal cord. But we also have spaces in our brain. So notice there's a space here which we call ventricles. Now, 2 of them are numbered. We have a 3rd and a 4th ventricle. So that kind of begs the question, which ventricles are 1 and 2? Well, that's these 2. So here's 1 and here's 2. But we just refer to them as the lateral ventricles, so we don't have to remember, the left or right, whether it's right, which one's 1 or 2. And so there are 2 lateral ventricles, which then flow into a 3rd ventricle, which goes around the thalamus of the diencephalon. So notice there are lateral ventricles which flow into the 3rd ventricle. So here's a pig brain forming. So notice the hollow nature of that. And then here, this is a mid-sagittal section. Notice you can see the lateral ventricles over the thalamus and under the cerebrum. Here's a model which would depict, the spaces that these things would depict. The 4th ventricle is under the cerebellum. The 3rd ventricle is along the midline, along the thalamus. So here you can see the lateral ventricles. There's the lateral ventricle. They would flow here into the 3rd ventricle, all right? And the space which you could see where the lateral ventricle flows into the 3rd ventricle is known as the interventricular foramen, sometimes called the foramen of Monro. And so the cerebrospinal fluid, which is surrounding the nervous system, helping to regulate its the ions, et cetera, supporting its weight. It flows from these lateral ventricles into the third ventricle. Notice that that goes around the thalamus at the midline of the body. And then from the third ventricle, it flows through this cerebral aqueduct, which goes through the midbrain. So if you remember, the midbrain has a tectum. That's the part which is dorsal to this cerebral aqueduct. So this would be the tectum of the midbrain, dorsal to the cerebral aqueduct. And then the rest of the midbrain, like the cerebral peduncles, would be ventral. So cerebrospinal fluid made in choroid plexus goes from the lateral ventricle to the third ventricle through the cerebral aqueduct to the fourth ventricle that's under the cerebellum. And from there, it can go into the central canal of the spinal cord, or it can then flow around through the space. If you recall that image of the meninges, so the dura mater is the outermost meninx, and then there is the arachnoid underneath and a subarachnoid space. So cerebrospinal fluid goes into the subarachnoid space, where it can be reabsorbed at these finger-like projections known as the arachnoid villi. Right, so here you can see the fourth ventricle, and from here cerebrospinal fluid could go into the central canal of the, so there's the fourth ventricle of the, you notice the cerebellum around it, and from there it could go into the central canal of the spinal cord, or in the meninges in the subarachnoid space, it could be reabsorbed. Reabsorbed there. I have another playlist which goes through the 12 cranial nerves. I'm just going to quickly review that, and once again, if you were interested, each individual video would have more information. As I'll mention when I talk about smell, we have these bipolar neurons in our nasal epithelium, which then send their axons through these olfactory nerves. We have lots of these olfactory nerves, which then go through the holes of the ethmoid bone. The ethmoid bone has a region known as the cribriform plate. And if you look, there's lots of tiny holes because as opposed to, like, instead of the optic nerve, we have these two big optic nerves. We don't have big olfactory nerves. We have lots of little ones, which then go through the olfactory foramina in the cribriform plate of the ethmoid bone. And this then goes into the olfactory bulb. I'll show you the olfactory bulbs in just a second. And from here, there would be an olfactory tract, which would then go to other regions of the cerebrum. I'll talk about smell, obviously, later, but smell is unique in many ways. Like, for example, it doesn't go through the thalamus. The olfactory bulbs used to be huge. So in fish, they're quite large. The cerebrum, once to a large extent, was a place of smell. And so a lot of organisms, a lot of mammals have fairly large, prominent olfactory bulb, whereas in humans, the olfactory bulbs are quite small. And so underneath the cerebrum, we would see an olfactory bulb and an olfactory tract. Also in the same general region, you would get this crisscrossing of the optic nerves known as the optic chiasma. You can see there in human brain, and here you can see in the sheep brain. Now, these cranial nerves are important, so important that students often have to memorize the cranial nerves. They're the number, so the olfactory nerve would be cranial nerve 1, the optic nerve is cranial nerve 2, but then there's lots of other things that you can memorize. And I'm sorry, just here's that cribriform plate of the ethmoid bone, and all of those little holes, that would be where those olfactory nerves pass. One could memorize the holes in the skull where the nerves pass, etc. So I have all the information in these videos. I'm just going to hit major points so that if you needed to know them in greater detail, say for lab, that you could refer to this. And so they are given numbers. Let me go through them, and at the end I'll talk about them in general and why they're important to know, etc. So the optic nerves are going to connect the eyes, and so the retina is going to send now axons, which then go through the optic foramina of the sphenoid bone. They mostly crisscross, so for the most part, not entirely, but most part, your information from your left eye is going to be processed in your right brain, and they'll be destined for the visual cortex of the cerebrum for the conscious processing. But remember that there are other involuntary centers, like the superior colliculus of the midbrain and the lateral geniculate nucleus of the thalamus. So when we talk about vision, we can get back into that. But there are the optic nerves, and after the crisscross, called the optic tract. I hate even saying this. Technically, the optic nerve isn't a nerve because a nerve leaves the brain. It's called a tract if it's connecting one part of, say, the brain or the cerebrum to another. Technically, if you look at an embryo, the retina, which now is part of your eye, formed as part of your cerebrum and then just moved. All right, and so this optic nerve technically is connecting the cerebrum to what used to be part of the cerebrum, the retina, just a long lost part of the cerebrum. So technically it's a tract rather than a nerve. As I'll get to, the reason that one wants to study this is because you can then help diagnose problems in patients based on where damage might be, for example. So visual deficits and even blindness could come with damage to the optic nerve. So those two nerves were sensory. All right, so what does the olfactory nerve do? Well, it allows for smell. What does the optic nerve do? It allows for sight and a couple other things related to vision. What does the eighth cranial nerve do? What used to be called the auditory nerve, but now called the vestibulocochlear nerve. It allows for hearing and other senses of the ear, like balance and acceleration. So some of these 12 cranial nerves, they are sensory. They are transmitting sensory information. Some of them are primarily motor. So for example, there are 3 cranial nerves, which are primarily moving the eye. The ocular motor nerve, the trochlear nerve, I'm sorry, and the abducens, I'm sorry, I drew a blank for a second. So the ocular motor, the trochlear, and the abducens nerves, they are responsible for moving the eye. And so if someone bangs their head or falls down and someone asks them, here, trace, can you follow this with my eye? You're actually checking 3 different cranial nerves. The ocular motor, cranial nerve 3, the trochlear, cranial nerve 4, and the abducens, cranial nerve 6. You're seeing how they're working, the nuclei where they originate, how they're working. And so, you're actually checking the functioning of 3 different cranial nerves. So here's, once again, just going back to the olfactory. Sorry, slightly out of order. But once again, from the olfactory bulb, going through those, the olfactory foramina in the cribriform plate. All right, so these 3 cranial nerves are motor, which are controlling the movements of the eye, the ocular motor, the trochlear, and the abducens. So if you're trying to memorize the 12 cranial nerves and what they do, so far you've got 3 which are primarily sensory, the olfactory, the optic, and the 8th cranial nerve, which does the senses of the ear, the vestibular cochlear. Why don't we do that one next? And then there are 3 which are primarily motor, moving the eye, the ocular motor, trochlear, and abducens. The eighth cranial nerve is sensory, and it is now getting the senses of the middle ear, which once again are, as we'll cover later, hearing, but then also balance and the sense of acceleration. Those are damage that would not only affect hearing, such as deafness and cause tinnitus, but also then balance issues and vertigo. You may remember that in the temporal bone, there's an internal auditory meatus. In addition to the external auditory meatus you see there, that's where that eighth cranial nerve comes out. Okay. So with my students then, if we're just trying to learn function, these next two, I ask them to think of them as being evil twins. And that's because being old, I grew up in the 80s, and every show right before they went off the air had an evil twin episode. So the evil twins, I think of cranial nerve 5, the trigeminal, which is a big complex nerve that actually comes from different nuclei in the brainstem. I like to play that off of the facial nerve because the trigeminal nerve allows you to feel the face but move the mouth. So therefore it's called a mixed nerve. So while some nerves are sensory or motor primarily, there are then mixed nerves which have both then sensory and motor portions. So the trigeminal nerve allows feeling the face, all right? So you feel the general senses of the face, touch, pressure, pain, etc. But then the movement of the mouth, so the control of the chewing muscles. So this allows you to feel your face and move your mouth. And that's different from the facial nerve, all right, which is cranial nerve 7, which allows you to move your face and feel your mouth. So by move your face, I mean facial expressions. And by feeling the mouth, I'm talking about most of your sense of taste. So your trigeminal and facial, if you want to remember them as being evil twins, the trigeminal allowing you to feel your face but move your mouth in chewing, whereas the facial nerve allows you to move your face, facial expressions, but then much of the sense of taste. Now, once again, this is part of a different chapter, but taste is actually a little bit more complex because there are different regions of our tongue which are perceiving things like sweet, salty, bitter, and then especially in children, there can be taste buds in the roof of the mouth and then even the throat. And so the facial nerve is getting the front of your tongue. But in the posterior tongue, like where we sense bitter, that's a glossopharyngeal nerve. But then the taste buds in the pharynx can actually be sending information along the vagus nerve. So while the facial nerve does a lot of taste, there are actually a couple of cranial nerves which perceive that. So there's a glossopharyngeal nerve. There's an accessory nerve, which is doing some neck muscles. But certainly one of the most important cranial nerves, certainly if you're going to study anatomy and physiology too, is cranial nerve 10, the vagus nerve. So cranial nerve 9 is a mixed nerve. Cranial nerve 10 is a mixed nerve, the vagus nerve. The reason it is so important is, say, in anatomy and physiology too, you're studying the guts, all right? You're studying, the heart, the lungs, the digestive tract, etc. Well, your brain wants to get information from your guts to know how you're doing, and then it wants to send commands to your guts. You want to control heart rate, blood pressure, breathing, etc. So this vagus nerve, to a large extent, is now getting information from the guts and then sending commands then to the guts. And so then in anatomy and physiology too, the vagus nerve is very important. And specifically, the vagus nerve is sending the parasympathetic commands. This is the rest and repose response to the guts, which would lower heart rate, lower lower breathing rate, stimulate the digestive tract, stimulate the immune system, etc. So most of the parasympathetic division of the autonomic nervous system travels through the vagus nerve. So cranial nerve 10, very important. The hypoglossal nerve can control, mixed nerve, but can control some tongue muscles. One of the things that I tell my students is, unfortunately, very often you have to memorize those the cranial nerves in order, simply because a lot of images, like you'll see in your textbook, you'll say, oh, cranial nerve 3 or cranial nerve 8, and you just have to know, cranial nerve 8, that's the that's the vestibulocochlear nerve. Cranial nerve 3, that's the oculomotor nerve. Cranial nerve 4, that's the trochlear, etc. And so, this is, something that a lot of students then just have to memorize. So therefore, what lots of students do is they set up an acronym that would help them with the OOO for olfactory, optic, oculomotor, trochlear, trigeminal, abducens, etc. There are a number of acronyms. I've certainly, heard the old one about, old Olympus top and a Finn and a German and the plants that they're smelling. There's one about a truck. However, I always felt, tell my students that, you need to remember those. And so the problem with the one about the truck or about the Finn and the German is that you might not remember them. And so there's the one that almost everybody uses, even though textbooks don't type it, which, involves, a sexual connotation. But once again, that also helps to illustrate, of course, if you're trying to remember something and you have a choice of an acronym involving a truck or a Finn and a German, and one which has, sexual innuendo, of course you're going to use the latter. Why? Because remember, it's parts of the limbic system, like the amygdala and hippocampus, which are encoding memories. We remember things which get an emotional rise out of us. And so if there's something that you would like to remember, of course, something that generates an emotion is going to be more easily remembered than something that isn't. And so, I go through this acronym a bit more in this video if you'd like. So the human brain is so incredibly complex. It is, without exaggeration, the most complex structure in the known universe. And I have tried in a series of lectures to then, break it into, parts and study, the brainstem, the cerebellum, etc. And so this has focused largely on the cerebrum. Some of these cranial nerves and their senses will come up later because, the general senses are covered in, a future chapter. So hopefully this has summarized much of the information on the cerebrum. But like I said, some of these videos will go through this material in greater detail.